receptor that is shortened. Those are the HER2 Herceptin patients who don't respond.
It's, I'm planning to talk about it. Predicting eligibility is the gatekeeper test for HER2. And like ER, it's a negative predictor. It's not a test that can tell you who will benefit from trastuzumab or lapatinib-based treatment. It's trying to tell who is so unlikely to benefit, it would be best to spare them the cost and the potential toxicity. So the primary test, which we call the eligibility test, is an exclusion test. It's a negative test. Once you have patients who are either 3+ or FISH positive, we now have a group of patients who may benefit. But why don't they all benefit? What are the tests we can do? And I was planning on talking about them, and I was going to talk about truncated receptors and other things like that. But rather than do that now, I'd like to get through some of this other background. So let's go for it. Here's the arrow of development. I think everyone also should know, I won't walk over there, but in 1983, Robert Weinberg, the celebrated molecular biologist at MIT, discovered a gene that transformed a rat neuroblastoma cell line that he called the NEU gene. Slayman showed that it was homologous to a gene that transformed breast cancer into an aggressive subtype, whether it was node negative or node positive, in the mid-'80s. And then in the early '90s, Genentech Corporation, at the prestigious address of one DNA Way, South San Francisco, California, did two things that shocked the pharmaceutical industry. Number one, they announced they were going to attempt to use the dehumanization technique that had allowed the multi-dosing of the anti-CD20 lymphoma antibody rituximab to achieve a recent FDA approval and growing clinical use, and do the same thing for a solid tumor. And investigators around the world said solid tumors were a bigger, let's call it, adversary than lymphomas. Lymphomas were driven by a single, translocation or some other genetic defect, and an antibody might work against those more clonal diseases. But against a multi-clonal, multi-pathway, complex malignancy like a solid tumor, antibody didn't have a chance. That was issue number one. But issue number two was even more serious. Genentech said, we got some preclinical and preliminary data here that this antibody we've developed won't work on every breast cancer. It'll only work on some of them. And we got a test in mind that would pick which those were. And we were going to segment the market to treating only the patients who had the test positive. In other words, rather than treat everybody and hope that nobody was hurt who didn't respond, but that we got paid for treating everybody, they were going to treat only the people they thought would possibly respond. And as you know from the data, if they hadn't done that, they wouldn't have an FDA-approved drug, would they? So the development went to the day in 1998 in December when the FDA, for the first time in its history, approved on two sides of the street in Gaithersburg, Maryland, an antibody therapeutic in combination with two chemotherapy regimens, either single-agent paclitaxel or adriamycin Cytoxan, that in its label was a test, the DACO Hercept test IHC, which must be performed and needed to be 2+ or 3+, that's how the label reads, in order for the patient to be eligible to receive the drug. And in the in vitro diagnostics branch of the FDA, they approved the DACO Hercept test, saying that this test was in the label of the trastuzumab drug and designed only to select patients as eligible for therapy. And there's only been one other drug test combination ever approved by the FDA. Anyone? Only one other. It's Erbitux and EGFR immunostaining in colorectal cancer. Which is ironic, isn't it? Because we're going to spend, after the break, a whole bunch of time talking about tests that really predict whether cetuximab or tocilizumab will work in colorectal cancer, not EGFR staining, but other tests like RAS mutation, et cetera. So really, right now, I'd have to argue with you that trastuzumab and HER2 testing is the only combination product ever approved by the FDA. A lot of people raise it. What about Gleevec and CML, for example? And the difference here is, although clearly that's a test drug combination, when the FDA approved imatinib, it did not specify what BCR-ABL test had to be performed. They left that open to the laboratory who was taking care of the patient. They could use various PCR methods and FISH methods, whatever they wanted. So they didn't regulate the test, they only regulated the drug. And I bring that up because I think the level of scrutiny for HER2 testing has to match how it was originally approved. Jim?
Can you comment on CISH regarding HER2 duplication rather than true amplification?
Sure. I mean, most of the patients who have HER2 overexpression at the 3+ level, probably 90+% have true amplification of HER2. A subset of 3+ staining patients do not have an amplification because the ratio of the HER2 gene copy number to the chromosome 17 probe is not above the cutoff, which today is 2.2. Yet, the numerator is above 4 or even above 6, and this is where we bring in the idea of chromosome 17 polysomy and extra copies on a mechanism other than amplification. It still remains controversial. I, for one, believe it's a dose relationship. If there are more numbers of the gene, there's no evidence they're ever silenced. They're not methylated. They do translate and they do transcribe. So polysomy exists.
So you don't think that's a relative contraindication for using CISH versus FISH with a subset?
No, actually the data is unbelievable in the concordance between CISH and FISH. There's been more than 20 publications now. I think the lowest concordance is 96%, and most are in the 97, 98. So it doesn't seem to be a problem. In my FISH reports, I never rely on the ratio. I know the FDA approval for the test is ratio-based, but I override that in my interpretation every time. First and foremost is the numerator. How many copies of HER2 are in this patient's average test result on 40 DNA copy numbers and 40 overlapping cells? If the numerator is above 4, the test will never be negative in my lab. If it's between 4 and 6, it's called equivocal. If it's greater than 6, it's called positive, independent completely of what the ratio is. Or another way to put it is never know only the ratio, because if you've got a ratio of, let's say, 2 in the equivocal range, and it's 4/2, that's not the same as 6/3, and it's not the same as 8/4. So pay attention to the numerator first and foremost. And so I have a much higher equivocal rate, and the ASCO CIP guidelines allow for this. They state that any 4 to 6 range FISH test is equivocal, not negative, even if the ratio is under 1.8. So ratio is a gimmick. It has limited, I believe, clinical value. Numerator, HER2 copy number, that's the test result you're really looking for. So I think I'll skip some of these background slides to speed up a bit. We're talking about the tests themselves.